Can start now.
Okay. Good evening, everyone. Today we are going to discuss about the diseases of wheat and mustard. First of all, wheat scientific name is Triticum aestivum, and the major diseases which attacks wheat are enlisted below. These are the fungal diseases, which includes black rust, yellow rust, brown rust, loose smut, common burnt kernel burnt, powdery mildew, flax mud, foot rot, leaf blight. And these are the causal organisms which causes the following diseases, which we will learn in the next slides. And these are few bacterial and viral diseases like bacterial leaf streak and barley yellow dwarf, barley yellow streak mosaic, and barley yellow striate mosaic. These are the nematode diseases like ear cockle, tundo or yellow ear rot, and molia disease. Now let's learn in detail. First of all, let's learn about stem or black rust. And the causal organism is Puccinia graminis triticum. It is a heteroceous rust, and the alternate host is Barbary, whose scientific name is Barbarius species. Heteroceous, the term heteroceous means uh, this rust has five spores, and it is macrocyclic, and all the five spores are occurring in the host plants, and the alternate host is Barbary, which is not present in India. So some volunteer hosts are there in which the pathogen survives. These are few symptoms like we can see reddish brown, dark reddish brown color pustules which occur on both the leaves, stems, and spikes. And these appear in scattered form, and sometimes they combine to form a collagen like structure. Prior to the pustule formation, some flakes also may appear. Before the spore masses break through the epidermis, the infection site feels rough to touch and give a ragged like tone appearance. These are the symptoms which can be seen through the eyes in fields, in the stems and leaves, and also in the spikes. These are the five spores I was talking about, due to which it is called macrocyclic fungi. So the stage zero was given at the last, so it was named like accordingly only, which is Spermagonia with spermatia and receptive hyphae. This is the pycnial stage, and the stage one is known as axial stage, which contains ascospores, and second stage is uradial stage, third one is teleal stage, and the fourth one is basidial stage. These are the spores of stem rust. The left one represents the uridospores, which are brown in color and oval shaped with four jump pores. And these are the right side. This is the teleospores, which is dark brown in color, is two-celled, bicelled. We can see two cells over there, and pedicellate and has a pointed apex. The tip, tip part is pointed, and the life cycle consists of the uridospores are the repeating spores which we can see on the stems and leaves, which keeps on repeating throughout the crop present in the field. And then when the summer ends, harvesting is done, it overwinters in the crop debris as in the form of teleospores, and then they germinate and form basidiospores, which infect the barberry crop, which is the alternate host for this fungi. And then again, spores are formed, which infects the wheat plant in the next season. These are the pycnospores and the spores. The survival and spread is the, as I said, the uridospores and teleospores are the repeating and resting spores, respectively. And these move. This is the airborne, long distance airborne fungi, which move from northern hills during off season on self-sown crops or volunteer crops to the southern hills. And the spreading is done through uridospores from hills. The favorable conditions are temperature should be 20 degrees Celsius to 30 degrees Celsius, and moisture should be 70 greater than 70 to 80%. Management can be done by mixed cropping, crop rotation, avoiding excess doses of nitrogen, sulfur dusting, spraying mancozeb and zinab or propiconazole at the rate of 0.1%. Growing resistant varieties like PBW343 and so on. Spraying 1 liter buttermilk plus 1 liter cow urine along with 8 liters of water. Next is the yellow rust or stripe rust, which is known as Puccinia. The causal organism is Puccinia staphylococcus. This is also a heterogeneous rust, but the alternate host is still unknown. We don't know about the alternate host till now. So the pustules are yellow to orange in color, which appears in narrow stripes, and the teleospores are also arranged in long stripes. That's why this rust is also known as stripe rust. These pustules can be seen on leaf sheath and leaves. These are the orange or yellow pustules. And the uridospores are yellow in color, and they are spherical or oval in shape with 6 to 18 germ spores. And the teleospores are dark brown in color, are bicelled and pedicellate, and are flattened from the top. And the life cycle is similar like the previous rust. First, we see the repeating spore, that is uridospore, which overwinters as teleospore, and then in the alternate or volunteer host, it in the off season it resides, and then again it spreads in the main season when wheat crops are sown. The survival and spread is also similar to the previous rust, as it is a rust fungi. And the favorable temperatures are 10 to 20 degrees Celsius, and the humidity is greater than 80%. Next comes the leaf or brown or orange rust, which is caused by Puccinia recondita. It is also heterogeneous, and the alternate host is Meadow rui, which is Thaliacrum species. The pustules are smaller than the stem rust, and they do not collagen, and they are orange or orange brown in color. The uridospores are, and they mostly appear on the upper surface of the leaf or leaf sheath, occasionally on the necks and awns also. And the symptoms are like this. On the leaves, there are brown or oranges brown color, and on stems also. And the pathogen is when under microscope we observe this pathogen, we can see brown or spherical structures which are known as uridospores, and the teleospores are two-celled and oblong, and they have round apex. And the life cycle is here also is similar. As we can see, the uridospores are the asexual and repeating spores. And then comes the teleal spore, which is the resting spore, which overwinters in the crop debris. And then when the favorable temperature and condition arrives, they germinate and form basidiospores and infect the alternate host, that is thalictrum. And then from there, asiospores come on to again to the wheat crop. And the survival and spread are similar as to the previous rust fungi. And the temperature is 20 to 25 degrees Celsius with free moisture. Then comes the loose mud, whose causal organism is Ustilago nuda triticum. This is an internally seed-borne disease, and the symptoms are the entire inflorescence, which is the main part of wheat crop, is covered with mass of olive black spores, except the rachis. And when wind blows, the whole spores are blown away and spreaded in the field, but the rachis is left back. And this appears, the head appears powdery, and the symptoms looks like this in the field. The rachis, except the rachis, whole inflorescence is converted to spore masses. And the pathogen under microscope appears as pale olive brown, spherical to oval in shape, and are adorned with minute echinulations. We can see some thorn-like appearance, which is called echinulations. And they germinate readily in water. Each spore produces 1 to 4 celled germ tubes, which is also known as promycelium. And the life cycle is those spore masses infect the whole field by blowing of veins, and then they are known as the teleospores. And when they reside in the seeds, they're internally seed borne, so they are reside in the seeds. And when we sow the infected seeds, they enter to the young seedlings, and when the flowering stage again arrives, the entire inflorescence is converted into spore masses. And the survival and spread is through veins, and the primary infection is caused by sowing infected seeds. Favorable conditions are cool and humid condition. Optimum temperature is 18 to 20 degrees Celsius with 65 to 85% relative humidity. The management is done by solar heat treatment or hot water treatment of seeds. When at that temperature, the fungus cannot survive, and we can sow the seed after that. And rogging out means removing the diseased earheads or plants from the field. Growing resistant varieties like Ras 2296 and Ras 6276 and etc. We can also use Vitavix at the rate of 2 gram per kg seed before sowing. Next disease is common bunt or heel bunt, or known as stinking smut. The causal organism is Tilletia triticum and Tilletia laevis. This is externally seed-borne, and the fungus attacks the seedling stage at 8 to 10 days old and becomes systemic, and from the seedling stage, they grows along with the tip of the shoot. At the time of flowering, the hyphae concentrate in the inflorescence and spikelets, transforming the ovary into smut sorus or dark green color. The disease plant mature earlier, and all the spikelets are affected. The stinking smuts are of two types, and the stinking name is given due to the rotten fish smell, which is caused by trimethylamine, which is produced by this fungus. And the symptoms are like this. The entire inflorescence appears like this. This is the infected one, and this is supposed to be the healthy one. And the pathogen under microscope, the Tilletia triticum appears reticulate and rough-walled, and the Triticum laevis appears smooth-walled and irregular shaped. The disease cycle is like this: when the infected head with burnt balls and the kernels are sown, the seedling along with the disease, it carries the disease, and it becomes systemic, due to which the teleospore and basidiospores are produced, and the conjugating sporidia with H structure is produced, which produces infectious hypha, and then it becomes systemic infection in wheat plant. And when the flowering stage appears, this mycelium invades the wheat head and then infects the whole inflorescence. The survival is through the spores of the seed surface germinate along with the seeds. Each produce a short fungal thread cluster of elongated cells, and these produce a secondary spore. And the favorable condition is 18 to 20 degrees Celsius with high soil moisture. The management is shallow showing and grow the crops during high temperature. Treat the seeds with carboxin and carbendazim. Resistant varieties can be sown like Kalyan Sona and the canal bunt, whose causal organism is Tilicia indica. This was first reported in Karnal in 1931, and the symptoms are not the ears are affected. Not all the ears are affected, and the grains are converted into sorai balls. The sorai are irregularly distributed, and on maturity, the spikes spread out and the bunted grains may be absorbed. These are the symptoms. This is how it looks, and this is the disease cycle. The diseased ear head, and when we take the grains and sow in the soil, then the spores are produced, they germinate, then sporidia and secondary sporidia are produced. And when again the flowering stage of the wheat crop arrives, they again infect the whole flower or ear head. Survival and spread, the infected seeds is the main source of infection as it is a seed-borne disease, and the favorable condition are again 18 to 20 degrees Celsius and the high soil moisture should be present. Powdery mildew, causal organism is Ericife graminis. We can see whitish powdery growth which appears on the leaf sheath or all over. We can see it in all parts of the wheat, like leaf sheath, stem, and floral parts. The powdery growth later becomes black in color, and the leaves also dry because the photosynthetic rate only reduces due to the blockage by the mycelial growth, whitish mycelial growth. And the symptoms are like this on leaves, leaf sheath, and inflorescence. And the fungus produces septate superficial hyaline mycelium. Hyaline means colorless with a short conidiophore. The fruiting body, sexual fruiting body is Clistothecia, which produces 9 to 30 asci, inside which ascospores are present. And conidia is the asexual spore, which is hyaline, single celled, and is produced in chains like this. These are the conidia which are produced in chains. And this is the Clistothecia, inside of which we can see the asci. This is three asci, and inside those, they were the ascospores present. And the disease cycle is, um, first of all, the infected part has developed Clistothecia on the lower leaf, and then it overwinters as mycelium on crop debris. And then in spring, the conidia and ascospore starts early infection. Mycelium develops on the young plants. Conidiophore are releases conidia, and again the infection appears in the wheat crop. Survival and spread, the fungus remains in the infected plant debris as fruiting body, which is the Clistothecia. Primary spread is through ascospores, secondary spread is through conidia. And this fungi favors cool and moderate temperature like 20 to 21 degrees Celsius and thrives well in high humidity. Thrives Okay, management is crop rotation, removal of plant debris, avoid excessive use of nitrogen, use resistant varieties, and we can spray wettable sulfur at 0 to 2% and carbendazim at 500 gram per hectare. Flag smut or stem smut or stripe smut. Causal organism is Urocystis triticum and Agropyri. The main uh, infection is from late seedling to maturity stage, and the seedling infection leads to twisting or drooping of leaves, and then withering is seen. The pathogen causes a systemic infection on host plant, forms a sori in vegetative parts, most commonly in leaves as a narrow strips between the veins, due to which a grayish black sori on the leaf blade or sheath can be seen. This is the twisting and curling of the leaf sheath, and these are the sori balls between the leaves. And the pathogen, this is how the pathogen looks under microscope. It is an aggregated form with 6 to 18 globules like hyaline structure attached to it. And the disease cycle is the sori, which is developed between the veins of leaves and glumes. That sori releases spore balls containing teleospores, and that overwinters in the soil or seeds, and then germinate. Germinating teleospore forms promycelium and sporidia, and when autumn and spring arrives, the sporidia again germinates and mycelium infects the seedlings. This fungus grows systemically with plants. Again, we can see the symptoms on the leaves and glooms. Survival and spread is through, as it is a seed or soil borne disease, and the smart spores are viable for more than 10 years, so it is unavoidable. And the primary infection is through infected seeds, and the resting spores are present in the soil. Temperature range is from 18 to 24 degrees Celsius. Relative humidity is 65% or above. Management, same crop rotation, removal of plant debris, seed treatment with carboxine, copper sulfate and copper carbonate. Uh, Resistant varieties like Pusa 44 and WG 377 can be grown. Foot rot, causal organism is Pythium graminicolum or Pythium aherinimanes. The symptoms which can be seen are the seedlings and roots and rootlets become brown in color, water soaked appearance is seen, and pale green, and have the seedlings are pale green in color and have a stunted growth. These are the symptoms. Rotting has happened, and the pathogen mycelium is well developed synesthetic without septation, and fungus have sporangia and zoospore as it is a lower fungi. The sexual spores are oospores, and this is how it looks under microscope. These are the vesicles present, and they germinate and produce sporangia. Sorry, they produce zoospores and oospores. Okay, these are the sporangia which we saw right now, and they germinate and produce oospores, and then zoospores are produced. Zoos inside a sporangium, zoospores are also can be produced. If favorable condition is there, then only oospore are produced, and these zoospores can cause infection in the wheat crop. Survival and spread is through soil and irrigated water. Pathogen survives in soil, and wet weather is favorable with high rainfall. Management is crop rotation, removal of plant debris, and seed treatment with apron. Leaf blight, which is caused by Alternaria triticina, Alternaria alternata, and Bipolaris sorokiniana. Symptoms are reddish brown oval spots which are arranged in concentric manner, appears, appears on younger seedlings with bright yellow margin, which is known as yellow halo. In severe condition, several spots collapse and cause drying of leaves. Survival is in the form of canidia. No. Okay, primary spread is through canidia, and is it survival is in seed. So it is externally seed-borne disease, and secondary spread is through airborne conidia. And favorable condition is the temperature is 25 degrees Celsius and relative humidity should be more than 70%. Or Geneve, seed treatment, bacterial leaf streak or black chaff, which is caused by Xanthomonas campestris, Pathovar undulusum, or Xanthomonas translucens, Pathovar undulosa. Initially, small linear water soaked areas can be developed and bacterial ooze can be seen. Later, lesions elongate and mix to form a narrow yellowish or brownish stripes. Spikes are blighted and turn black. Cream to yellow colored slime or droplets like ooze are seen. These are the symptoms: bacterial leaf streak, and these are the ooze. This is how we can see when it is cultured on the lab, and this is under the microscope. Pathogen is a proteobacteria and is gram-negative and non-spore forming bacteria. It is motile, as we can see in the previous slide. It has a single polar flagellum. It can grow in temperature range of 4 degree to 37 degrees Celsius. Survival is in seeds, crop residues, or in soil. Transmitted by splashing water, plant to plant contact, and insects. Volunteer and grassy weeds are also the source of inoculums. Favorable condition is wet weather and irrigation water and rainfall. Temperature is 25 to 30 degrees Celsius. Management is by crop rotation, removing the volunteer and grassy weeds and infected plants, avoid or over irrigation, use of disease free seeds and resistant cultivars like Blade, Cromwell, Faller, Howard, and Knudson. Then the mustard, and these are the list of diseases in mustard. The fungal diseases like white rust, alternaria blight, downy mildew, powdery mildew, club root, and sclerotinia stem rot are the major fungal diseases. Bacterial like bacterial blight and viral diseases like mosaic can be seen in mustard crop. White rust is the major crop which is caused by Albugo candida. The symptoms are both local and systemic. In local infection, like we can see on leaves, white to creamish yellow pustules which appear on the leaf. Later, it collagens to form patches. In systemic infection, when humid weather arrives, white rust and downy cause swelling and distortion of the stem and floral parts, due to which hypertrophy and hyperplasia occurs, and a stag head structure can be seen in the inflorescence. Uh, this is the stag head structure I was talking about, and these are the pustules, whitish creamish pustules on the underneath of the leaves, which is the localized infection. And the survival and spread is survives through oospores in affected host tissues and soil. Secondary infection is carried out by sporangia and zoospore. Favorable condition are moist relative humidity and warm weather and intermediate rains. This is how it looks under microscope. These are the sporangiophore, these and these are the sporangium. Mm. Okay, the management are using resistant varieties, use at least a 3-year crop rotation, removal of weeds, Seed treatment with metalaxyl at the rate of 6 gram per kg, spraying Bordeaux mixture or dithinet or Ridomil. Alternaria blight or Alternaria leaf spot. Causal organism is Alternaria brassicae or Alternaria brassiciola. Symptoms are small circular brown necrotic spots which are concentric in nature, cover large patches showing brightening and defoliation in severe cases. This can infect stem, pod, which are elongated at later stage. Infected pods produces small discolored and shriveled seeds. These are the concentric ring structures I was talking about on the leaves, and the yellow halo can also be seen. These are the necrotic spots. And this is how it looks under microscope. Muriform conidia, which is the prominent structure of Alternaria species only. They form muriform conidia, which is muriform means two types of septations are present, one longitudinal, one vertical. And this is the beak part of the conidia. When spots appear, dark brown or black leaf spots, and they produce a muriform conidia. These infects the pods and seeds also, and when these seeds are sown, they again carry the disease to the plant. Spots on the seedlings and then again on the main crop. The survival is on the seeds, as we saw right now, and spread is through conidia or mycelium in diseased plants, conidia or mycelium. And favorable condition are moist relative humidity with warm weather. Intermediate rain favors disease development. Management are crop rotation, removal of infected plants, using of healthy seeds, seed treatment with Trichoderma hazarianum, spraying mancozeb or Ridomil. Downy mildew, which is caused by Peronospora parasitica. The symptoms are grayish white, irregular, necrotic patches on the lower surface of the leaf. Latter, when favorable conditions are seen, brownish white fungal growth may be also seen on the spots. Survival and spread is through survives through oospores on the affected plant tissues or weed hosts. Favorable conditions are 10 to 20 degrees Celsius and relative humidity is greater than 90%. These are the whitish growth on the underneath of the leaves and chlorotic patches on the above side of the leaf. This is how it looks under microscope. These are the sporangia and dichotomously branched sporangiophore can be seen. Management is through crop rotation, removal of infected plants, avoid over-irrigation, spraying mancozeb or chlorothianol or oxathiopropinil. Powdery mildew caused by Ericife polygony. Whitish powdery growth, mycelial growth can be seen on the upper side of the leaf. Later, when infection is severe, we can see chlorotic patches when the powdery masses are removed. This is how it looks under microscope. These are the chains of conidia formed. We earlier also discussed about these ericife in wheat crop, which causes powdery mildew in wheat. These are the clistothecia, which is the fruiting body, inside which ascus are present and ascospores are also produced. Clubroot or finger toe disease caused by Plasmodiophora brassicae. Symptoms are plants are stunted, tiny nodules to large club-shaped outgrowth in roots. Leaves are pale green or yellow, followed by wilting. And survival is in soil as resting spore. and this acts as primary source of inoculum. This is the symptoms. It looks like foot and toe, so they have named it as foot and toe disease. And this plasmodiophora is not a real fungi. Uh, does not As it does not produces mycelium. The body is like multinuclear plasmidial like. That's why its name is plasmidiophora. We can see the structures under microscope. These are the resting spore which produce zoospore, which infects the plant and the roots, and then again the plasmodiophora invade cells and cause gall formation. Club root disintegrates, releasing resting spores, and then again the resting spores produces the zoospores, and this is how the disease cycle of plasmodiophora looks. Favorable condition are humid weather and high soil moisture. Management is through removal of plant debris, soil solarization, using of resistant varieties, using susceptible plants like mustard, radish, turnip. Sclerotinia stem rot, which is caused by Sclerotinia sclerotiorum. Symptoms are elongated water-soaked lesions on stem, covered with cottony mycelial growth. Plant looks like whitish from distance. Premature ripening and shredding of stems. Brown to black sclerotial bodies may also be seen. Due to the whitish mycelial growth, it is also known as named as white mold or snow mold. This is how it looks, snow-like appearance or whitish mycelial growth. And when we split the stems of this plant, we can see sclerotia like black mustard seed-like structure inside the uh, vascular tissues. These are the sclerotia which are produced. And the fruiting body is apothecia emerged from sclerotia located near the soil surface. Then ascospores are released and blown by the winds. These ascospore infects the aerial part of the plant. Lesions develop and expands, and then again maturation of sclerotia on and in diseased tissues like we saw in the vascular bundle, and then sclerotia accumulate in the soil again on decay matters. Then sclerotium overwinters, and then again apothecia is emerged, and then sclerotia is released. When we culture it in the lab, this is how the mustard seed like sclerotia are seen on the corners of the Petri plate. This is the apothecium, and they are released. The survival is as mycelium in dead or live plants, as sclerotia in infected plants, or in the soil surface with seeds as contaminant. Favorable conditions are high humidity, which is 90 to 95%. Average temperature is 18 to 25 degrees Celsius, and wind favors the disease development. Management is again crop rotation. Avoid planting next to field that has severe sclerotinia in the past 4 to 5 years. Plant thoroughly clean seeds. Avoid dense stands of canola. Bacterial blight or bacterial black rot caused by Xanthomonas campestris, Pathova campestris. Symptoms are yellow and chlorotic spots, which forms a V-shaped area with base of V towards the midrib. And the vein shows brown to black discoloration. Midrib cracking can be seen, browning of veins, withering, dark colored streaks. Stem become hollow due to internal rotting. In severe cases, vesicular bundles of the stem also turn brown and the plant wilts. This is how the V-shaped chlorosis is seen from base to the midrib, towards the midrib. Survival is in infected plant residue. Pathogen spreads by soil and irrigated water. Favorable conditions are warm and humid climate. Management is through seed treatment with mercury chloride for 30 minutes. Hot water treatment at 50 degrees Celsius for 30 minutes. A 3-year crop rotation can be done. Strict sanitary precautions should be maintained. Thank you. Any questions?
Thank you, Ms. Lakshmi Mohanty, for the presentation. Now I'll take up some questions. Are you able to
Yes,
hear
ma'am.
me properly now? Am I
Yes,
audible
ma'am.
properly? Yeah, okay. Okay, 1 second.
Okay, ma'am.
Just 1 second. Okay. So first question is, what are some common diseases that affect wheat and mustard crops?
In wheat, we can see rust
Hello? Hello?
diseases like yellow rust arrives first, and then brown rust, and then black rust. And then smut diseases also can be seen. Sorry, ma'am.
No, please go ahead. I was not able to hear you for a while, but you are clear
Okay.
now.
Yes, ma'am.
I'm able to hear you now.
Ma'am, you can hear me?
Yes, I can hear you. Please proceed.
Yes. Like I was saying, in wheat crop, mostly we see diseases like rust and then smut, black smut, blue smut, which is caused by ustilago, and then kernal smut, canal burnt, common burnt, powdery mildew, and in mustard we can see like downy mildew, powdery mildew, alternaria leaf spots, and then white rust.
Okay. And how do fungal pathogens like rust and powdery mildew impact wheat and mustard plants?
Okay, like in powdery mildew, the pathogen is Ericife species, in which clistothecias are produced on the other underneath of leaves at the latter stages, and these clistothecia has ascus inside them, which produces ascospores, and these ascospores spread the infection to the further plants. This is how powdery mildew is spread.
Okay. And what are the symptoms of wheat rust and
The
how
sec-
does it affect crop yield?
okay, wheat rust. In wheat rust, there are three types of rust. First one is yellow rust, in which the leaves are covered with yellow pustules, due to which photosynthetic rate is lowered, and which will indirectly or directly affect the yield production, grain production mainly, because we know that photosynthesis, due to photosynthesis only the grains or fruits are developed, and when photosynthesis is reduced, then they cannot healthier seeds cannot be produced. Similarly, stem rust or brown rust and black rust are uh, reducing the crop yield of the wheat crop.
Okay. And next question. Can you explain the life cycle of a fungal pathogen that affects mustard crops?
Okay. Uh, I will I can talk about like we discussed about alternaria. In alternaria, we saw concentric ring-like structure on leaves, which which produces muriform conidia, like that muriform conidia structures we saw, and that spreads the infection to the other plants. And these are when the plant is harvested, these are stored in the leaves and plant debris, and they reside there, and when the next season arrives, they again uh, germinate and produces and causes infection in the alternaria, mustard plants.
Okay. What are some strategies farmers use to prevent or manage diseases in wheat and mustard crops?
Okay. First of all, we can break the chain of, like, mostly we grow rice and wheat, which are both belonging to the grasses family, like poaceae family. So we need to break that cycle. Otherwise, these diseases will be prevailing. The farmers mostly use chemicals like mancozeb, plantavax, vitavax, and seed treatment with captain or therum. So which is like which is now we can see ki, it is improving, which is not infecting our crops, our disease free field can be seen. But on latter stages, due to the chemically resistant, the pathogen will develop a resistance, and in long run it is not like helpful. Some of the chemicals are banned also due to the over excessive use of these chemicals. So we need to break the cycle of rice and wheat. We can grow any leguminous crop or other family crops, which will break the pathogen cycle also, and we can control the diseases also.
Okay. And how do viral diseases like wheat streak mosaic virus affect wheat plants?
Leaf streak mosaic virus, uh, like it is not the major disease, but as we know, viral diseases are transmitted through some vector, um, and there may be some vector um, which is helping in the transmission of the diseases from one plant to another. Otherwise, like, contaminants are there, like if we use a sickle in one plant which is having the viral disease, and then we use it on the another plant, then the virus is easily transmitted to the other plant.
Okay. Are there any resistant varieties of wheat or mustard that are less susceptible to diseases?
uh, no, ma'am. Actually, resistant varieties are mainly developed to for the resistance of the diseases. Like if they are susceptible, then why will they release? The seed certification unit only will cancel the seeds because they are susceptible to some diseases. So resistant varieties are not susceptible to, like, the crop diseases are prevailing. Mostly the microorganisms are developing resistance to the chemicals, due to which we need to develop more resistant varieties.
Okay. Okay. And what role does crop rotation play in reducing the incidence of diseases in wheat and mustard fields?
Okay. When we do crop rotation, like if you are growing wheat, which belongs to Poaceae family, which is a grass family, so we can do crop rotation with some leguminous or solanaceous crops, so that we can break the disease cycle which is helping the disease to reside in plant debris or some soil, or in as a contaminant or some spore. So we can break that cycle by planting some other family crops, if we can grow field pea or some leguminous crops, that will help us to fix the nitrogen also, and also will help to break the disease cycle which is continuing from years.
Okay. And how do environmental factors contribute to the spread of diseases in wheat and mustard crops?
Okay, like we know, these crops are mostly grown in Rabi season and harvested during this time of the year. So these crops, most these diseases also mostly we saw they like warm cooler climate. Some diseases are there which like warmer climate, but mostly they like cooler temperature, the diseases. And water is the main, if rain occurs or some dew are there, they helps in transmission of the disease. So like these are the environmental factors like dew, rain, and through rain splash or water, the spores are being disseminated to the other crops or other fields.
Okay. And last question. What are the economic impacts of diseases on wheat and mustard production?
Economic impacts.
Yes.
Okay. Like I said, they will reduce the crop yield. Like in wheat, we have main economic part is the grains or seeds, which we take from the plant. If the disease is infecting, then in loose mud or loose mud, the whole grain is converted into sorrel, like spore masses, and on smut diseases, whole grains are converted into sporous sori balls, due to which heavy yield loss is seen. And in mustard also, they infect the seeds, the pods of the plant, and due to which the seeds are infected, and we cannot use it to market in the marketing for those seeds in marketing, because those are infected already, so people won't buy it. So the economical importance will decrease also, and the yield is decreasing due to the disease, infection of disease in the plants.
Okay. 1 second. Okay, Miss Laxmipriya, I don't see any more questions. And before I end, I would like to ask one last question. Please let us know a little about your career background and what made you to take an interest in that particular subject, especially agriculture field. How did you enter?
Okay. Okay. Actually, I'm a student of BSc Agriculture in LPU, and as a agriculture student, the most important and what is what is affecting the crop yield. The major factors are like diseases or pests or something. So, as an agriculture student, our main concern is how to increase it, how to minimize the diseases or minimize the pest, so that we can increase the yields. So and this pathology, this is a subject which not everyone chooses, but I like this subject particularly because I'm when I see the diseases, I am more interested into it, that what is it and how the spores will look, how they are infecting, how they are causing losses, and all those things, they like attract me. That's why I developed interest, and then now I'm just reading. Now I'm not that expert that I can say everything, but yes, I am interested in this subject. And as an agriculture student, pathogens and diseases are the main part of our course.
Okay, okay.
Thank you, ma'am. Can I
Okay,
close
Ms.
the meeting?
Laxmipriya, yeah. We have now come to end of question round. On behalf of agricultureinformation.com, we like to thank you for the detailed presentation and answering all the questions. And we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Okay, ma'am. Thank you.
You're most welcome. Thank you.